Also GATE 2022 Life Sciences, Section P Biochemistry, question number 37. Two RNAs shown below were used separately as a template in an in vitro translation system, okay, which can generate proteins in all possible reading frames. This is very important, all possible reading frames, different possible reading frames. These are the two RNAs, this is RNA1 and this is RNA2, and these are the sequences of the RNA templates. The RNA1 translated product contained arginine and glutamate, and the RNA2 translated product contained arginine, glutamate and lysine. Which one of the following codons directs the incorporation of arginine? And the options are A, AAG, B, AGA, C, GAA and D, GAG. Okay, so we all know what is in vitro translation system. So in in vitro translation system, what we do, we'll have a ribosome, eukaryotic or prokaryotic ribosomes, whichever translation system we are using. Okay, so in lab we generally use, we use a wholesale extract from either plant source or an animal source as a translation system. So that wholesale extract will contain all the ribosomes, it will contain all the machineries which include ribosomes and tRNAs, right? What all things we require? We need first ribosomes, tRNAs, amino acids, right? We need amino acids also, and all the enzymes, these are the all. Apart from this, we also required miscellaneous things like cofactors, ions. So we have this in vitro, so we can do this in vitro translation. So it could be either synthetic, the system could be either synthetic, that is we can put in all these things in a test tube and do the translation, or we can use a whole cell extract, we can directly use a cell extract. Okay, the cell extract either from an animal source or a plant source will contain all this required all these requirements, like essential requirements will be included in the cell extract, or we can use a. So now to do in vitro translation, first we need RNA. So these two RNAs are given here. So they have a reading frame. So what is reading frame? For example, I'll take the RNA1 example. So RNA1 has AG n times. So the AG will repeat n number of times. Okay, AG will repeat in n number of times like this. So they have given us this direction. Okay, the direction is also important. So always remember the direction from which the RNA template is read is from 5' to 3' direction. Okay, it is always read in 5' to 3' direction. So the AUG sequence will be at 5' end. Okay, always remember this. So here the first RNA sequence is also, they have given us in 5' to 3' direction. Okay, and the sequence is AG n time. So they have said that these are the two templates, and we need to find out which amino acids are getting generated. The product contains which amino acid. Okay, so this can be one reading frame. So reading frame, as you know, it should contain 3. The reading frame, the code is a 3 base code, right? We all know that Marshall Nirenberg has found out that the reading frame is a 3 base code, right? The 3 bases of the mRNA will code for one amino acid. So this is one reading frame, right? So here we can see there are two different sequences. So in the first reading frame we have AGA and GAG. These are two different 3-letter codons, right? These are the codons. So AGA could be either arginine or glutamate. Similarly, GAG could either be arginine or glutamate. We don't know that as of yet. Okay. So the first reading frame we are getting these sequences. Okay. Now the second reading frame will begin from here, GAG, AGA, GAG and AGA. Okay, so as you can see, in the reading frame 1 we are getting AGA and GAG. In the reading frame 2 we are getting GAG and AGA. Okay, so we are getting only 2 amino acids, right? Here this will code for 2 amino acids, which is arginine and glutamine, and the second reading frame also we are getting 2 amino acids. Okay, and the third reading frame, do we have a third reading frame? AGA and GAG, which is third reading frame is also same as the first one, AGA and GAG. So if the ribosome will recognize this AUG, I have again AGU, AGU. Okay, this is just an example. Okay, a simple example to why the reading frames will be different. Okay, so if the ribosome will recognize this AMG, this is the first this is a AUG. If it recognizes, so this is one amino acid, this will be my second amino acid, right? This will be my third amino acid. Okay, now as you can see, this third amino acid, it is considering GAU, right? It is considering GAU, and this AUG sequence will not, the ribosome will no longer recognize this GAU sequence. Okay, and the next sequence will be GAG. Okay, fifth amino acid will be UAG. Okay, so what will happen if the ribosome is recognizing here at this sequence? If ribosome is recognizing, so this will be our first amino acid, then we'll have AGU, then we'll have AGU. So this peptide will be different. The peptide translated from this ribosome will be different, and peptide translated from this ribosome will be different. Why? Here we'll have a different amino acid. These are the two reading frames. Okay, there will be also third reading frame if I have a AUG here, and we'll have a completely third different reading frame. So since these are three codons, okay, each amino acid codes for 3 codons. So what will happen? We'll have a special amino acid if we start from A. We'll have a different amino acid sequence if we start from B. We'll have a unique amino acid sequence if we start from A. But what will happen in the fourth? The fourth amino acid sequence will be same as this A. The only difference will be it will not have this first amino acid. So the AGA, if I start from AGA, GAG, AGA, GAG, this is my amino acid sequence. Okay, this is, for example, let's consider this is arginine. The 2 amino acid sequence will be arginine, glutamate, arginine, glutamate. Whereas in the second reading frame, the amino acid sequence is glutamate, arginine, glutamate, arginine, right? Because it is starting from GAG. right? It is starting from GAG. The third amino acid sequence will start from AGA. So it will be again arginine, glutamate, arginine. Since it is a 3-base sequence for an amino acid, that's why I said there are 3 possible reading frames. So RNA is a single stranded, so it will have 3 possible reading frames. Okay, so I'll write this on next page. So our first RNA sequence was AG n times. Okay, so for at 5 prime we have AG, okay, 3 prime. So the first reading frame is AGA and GAG. Okay, and this will continue AGA, GAG till n times, right? This is first reading frame. The second reading frame will start from second nucleotide. Okay, so the first step is to find this 3 base set, write it down, write the sequence, then from that 3 base set, select the second codon, again make a 3 base set which is GAG, then AGA again, and this will go on. And the third reading frame is same as the first, AGA, GAG. Okay, and this these amino acids either can be glutamate or arginine. We don't know, we have to find out. The question is that we have to find out which is arginine and which is glutamate. Okay, so in the second RNA sequence we have AAG n times. Okay, and this is also from 5 prime to 3 prime direction. So we have AAG repeating, AAG, so again we'll select this 3 base and we'll write. So here obviously it is a 3 base, so AAG will repeat n times. So first reading frame will be AAG n number of times, right? The second reading frame will be AGA n number of times, and the third reading frame will be, this is the third, right? This third base, 3 nucleotides, GAA n number of time, right? GAA, right? And here the amino acids are arginine, glutamate and lysine. So can you see the common, which is common in both? Which amino acid sequence is common in both?
AGA.
AGA, right? AGA is only amino acid which is common in both, right? Do you see any other amino acid which is common in both? No. So remember, AGA is common in both. So as you can see, so AGA is coming common in both. So the correct answer is B, right? The correct answer is B. And if you can see the 3-letter sequence, the 3-letter amino acid, arginine is coded by 6 different codons. So AGA is also coded by arginine. Okay, So the correct answer is B, AGA.